Let's assume the rifampicin takes the shape of this in 3 dimensions. Okay? Where it can go and bind? So it can go and bind to other proteins, some part of the ribosome, a cleft. You know biology, right? Can bind. It has to find a surface, a specific, right? Very good. Now what about this? Do you think this will be very specific to binding somewhere? No. It is too uniform. The shape is, right? It depends on the charge, but it is already becoming a little bit difficult. It has a difficulty. Now let me make it bigger. Now it has some cavities, some sharpness. Now will it bind? It will bind, I can tell you. Because if you put this many atoms, I give you rifampicin as this size. So this is rifampicin. So this is much bigger than rifampicin, more than 500, okay? What do you expect? It will have many shapes and charge complementary surfaces which can go into the cleft and cavities of various biological systems. So it will bind, but bind very high non-specificity and non-selectivity. Correct? Now, let us understand the specificity and selectivity of this plug. Let's see if I remove this. Will it lose its specificity? 2-pin plug. It can plug this way, it can go this way. Right? Now by 3-pin, I brought specificity. Right? Now, will it bind? Will it go into a plug point which is 2-pin? So no selectivity. So therefore, look at old houses. Look at here. You see, no, this is the ultra modern. Okay? If you go back your old home, you will see everywhere 2-pin plug point. So selectivity and specificity is one at the cost of the other. Correct? The dream molecule is the one which will exactly go and fit and will leave rest not there. So now this is the famous Koshland model of 3-point attachment, on the basis of which all selectivity of enzymes in chemistry takes place. That's why 3 residues, 1 hydroxyl, 1 amine, and 1 carboxyl group. What is that? Aspartic acid, histidine and serine, active site. This you know, biologist, because you are majority biology. Huh? Serine proteases. Am I right? Serine proteases has their active site, serine, histidine, and aspartic acid. So carboxyl group, histidine, the NH, and the carboxyl from and the OH from the serine. So it is all 3-point. Therefore, you can see how difficult for you to design a drug which will go into the active site of an enzyme. Because enzyme active sites are like this. Everything will bind. Very difficult. So therefore, you have to design differently. Now, okay, so is that the bottleneck? My argument is yes. We haven't been able to understand the system to discover chemistry of the life and to make discovery. Once we make discovery, whoop, then today's economic times, you will see there's a series of companies, right? The whole economic times has series of companies who can take your discovery molecule and make its cheapest drug in the world. Okay? Those who haven't seen this, I suggest that you look at this table. It starts with the highest sell, Cipla, Sun Pharmaceutical, Cadila, Workhard, Torrent, Biocon, Jubilant, Panacea, Glenmark, you just name it. Daga, 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 daga. All of them don't know how to discover. But if Astra has discovered, if Pfizer has discovered, they know how to copy. Fat. And then they know very well how to make it cheaper and cheaper and cheaper and bring it to the market through the all regulatory processes and provide it at the cheapest level. You know, when Rentec was discovered, it was a billions of dollars product. Right? Today, you buy 20 Rentec tablet in 10 rupees in India. 50 paisa. Right? That's the power of the generic.